So good afternoon, everyone. My name is Dalia Haydar. I'm one of the CCF Scientific Advisory Board members and one of the faculty at Children's National Hospital here in DC. We had an amazing morning session and 2 wonderful poster sessions, and I'm so excited to kick off the afternoon session with you. To start with, I would like to introduce Dr. Challice Bonifant. Actually, Challice and me descend from the same car training tree. So I'm so excited to to learn more about some of her car NK work.
Thank you all. I'm so excited to be here and have to confess that I originate from the DMV. So I grew up in Martinsburg. I went to medical school at Georgetown, and during my graduate training, Dr. Turetsky let me come to his pediatric oncology clinic, which really lit a little bit of a fire. I spent a couple of rotations at the POB, and I also went to DC Children's. So here I am now at Hopkins. So a little piece of everyone is inside of me. Thank you. As Dalia said, I am going to be talking about CAR NK cells and really NK cells as anti-cancer immunotherapy. So just for a broad overview, NK cells are amazing, and I'm also going to touch on some clinical challenges and then talk a little bit about how in the lab we're trying to solve some of those challenges to ultimately get to clinic, where we know deeply as clinicians it really matters. So why NK cells? I'm not sure I saw any NK cell posters that weren't from people in my lab, but I did see a lot of T cell posters. And NK cells are really, really important because they can be given from healthy donors to patients. So there isn't HLA restriction like there are with T cells in terms of safety and infusion. And so what this does is break down some of the barriers that lead to increased costs, lead to manufacturing complexity, and also, very importantly, lead to variability in product. So a single NK cell therapy could potentially be expanded from a single donor for an entire clinical trial that then would allow us to learn something about the therapy itself and not necessarily think a lot about donor-to-donor variability. Secondly, NK cells have innate killing activity, and so my disease of choice, what I have been working on really now for decades, is pediatric AML. AML is very heterogeneous. Solid tumors are as well. We heard this morning a bit about sarcomas, brain tumors. These are diseases in which the cells that make up the disease as a whole have variable expression of proteins on their surface. So CAR T-cell therapy or T-cell therapy is really restricted by activation through binding a single target. NK cells are not that way. So here's a cartoon that I stole from one of my lab members that shows very nicely a number of the germline-encoded receptors that are expressed on the NK cell surface. They're inhibitory receptors and they're activating receptors. Transformed cells, or cells that are generally stressed or infected, upregulate their expression of activating ligands that then can activate NK cells to recognize and kill those cells, independent of whether or not they're expressing a specific protein. So NK cells can then, we hope, that cytotoxicity can be enhanced or localized by expression of a chimeric receptor or some other modifying feature, but in general, they have the capability to sort of clear heterogeneous disease. And then sometimes we talk about this in the positive context, sometimes we talk about it in a negative context, but NK cells themselves have limited persistence really compared to T cells that have been shown to persist for decades in patients. And so this is important if you end up kind of having some off-cancer on-target or even off-cancer off-target systemic toxicity that's limited by the lifespan of the cells themselves. So again, just to highlight some of the things that are really limiting in terms of T-cell based immunotherapies are not limiting for NK cell therapies. So I mentioned my disease of focus has really been AML, or acute myeloid leukemia, and that is because there is very clearly remains great clinical need for patients who have high-risk relapse or refractory disease. These patients receive intensive chemotherapy and often stem cell transplant, and yet around 40% of them still relapse, and once they're relapsed, it's even more challenging to treat if we can treat them at all. So what do we know about NK cell activity against AML? Over the past couple of decades, there have been a number of clinical trials that have shown us, just kind of in sum, that NK cell infusions are safe. These trials have a variety of different products where the NK cells were sometimes collected from the patient, sometimes collected from a donor, sometimes collected from sort of a third-party healthy donor, and they were treated in a variety of different ways in terms of the isolation and the ex vivo expansion. And so it's really hard to say precisely we know what NK cells will do in patients who have AML. But that being said, there have been hints of activity and hints of really significant anti-tumor activity that is very encouraging and stimulating us to continue on. Highlighted in this table are 3 of the more recent trials that have included NK cell ex vivo expansion, important to us because that's something that we are often doing in the lab, and that have shown also really nice anti-tumor activity in the face of a really challenging to treat AML. So why do I care? I think I mentioned I have focused on AML just because there is a great clinical need. What is here on the right-hand side of the slide is data from originally published by Heather Simons, who put it together, looking at the patients treated with what is essentially our standard of care at Johns Hopkins for high-risk AML, which would be a myeloablative conditioning, or MAC, which is very high-intensity chemotherapy paired with a half-matched stem cell transplant used as treatment for AML. So the drugs and sort of the treatment course are on the left, and then on the right, outcomes that were published in 2020 showing that the relapse incidence in these patients is around 40%. So 60% are cured, let's celebrate that, but still 40% are not. Also, data from Hopkins, I always advocate for doing really kind of deep correlative biology regardless of clinical trial so that then we can learn something even if the trial isn't as successful as hoped. So this is some correlative biology data looking at adult patients, so from our adult colleagues, treated also mostly with reduced intensity, but also some myeloablative conditioning and transplant for AML, showing that quantitative numbers of NK cells that are circulating in the patients a month after the transplant can predict relapse. So essentially, if you have more NK cells, there is a less incidence of relapse. So with that being said, there are a couple of key questions that are really centering what we're talking about in my group and what I'm trying to advocate for the field, and you all hopefully will go home and can't sleep because you're thinking about these questions too. But first, if we're expanding NK cells, so if we're taking them, expanding them up to a dose to infuse them to patients, are we changing the characteristics of those cells such that they no longer have an activated or cytotoxic phenotype? And then second, we know from now, since my fellowship when we were doing very early phase CAR T cell studies, that putting a CAR in a T cell changes a lot. And for hematologic malignancy, this makes a big difference if it's ALL or multiple myeloma. Not sure for AML that we've seen these great successes. Maybe if we put the CAR in an NK cell, that's how we will change things. But then does it even do anything more than the activating receptors on the NK cell itself? Okay, so to the first question. Megan Zinsky here in the audience is, I'm laughing because I asked her a couple of days ago, can I please steal all of your slides for this presentation I'm about to give? She is a third-year fellow who very sadly for me and happily for her is moving on to OHSU in 3 weeks for a faculty position. But essentially came to the lab not knowing much about NK cells, not knowing much about lab work at all, and then was able to really dig deep into a complicated project and get really, really detailed and robust data that then we're building upon and will continue to do so in the future. So to the first question, what happens when we activate and expand NK cells ex vivo? So there are a number of different ways that people activate and expand NK cells ex vivo. We, in the lab, prefer to use feeder cells, irradiated feeder cells, to get a really strong proliferative signal right off the bat that then allows the cells to expand up to a really nice dose in a week to 10 days. There are different feeder cells that have been used clinically, and the differences really relate to the expression of what is on the feeder cell surface. And the feeder cells that have been used clinically all express 4-1BB ligand. So it's not displayed in this, but they're K562s that are irradiated, express 4-1BB ligand, and then also usually express either IL-15 or IL-21. More recently, there are clinical grade, but NK cells that have been used for clinical trial that have also been expanded in the presence of what we otherwise understand to be an immunosuppressive cytokine, TGF-beta. But these cells retain activity, and the intention was that they would be active in a TGF-beta rich environment, which is in many solid and brain tumors. And so Megan set about to compare these 3 expansion conditions to really understand a bit more about maybe which one was better, what was the phenotype of the NK cells, and then to complement some of the data that already existed with the TGF-beta sort of imprinted NK cells. I'm not going to steal all of her thunder. Her poster is now down, so hopefully you all were able to see it from her. But this is just showing broadly across some of her data, looked at a number of different expansions, a number of different NK cell donors, but then aligned with how they were expanded and then collected on day 10 of that expansion. Look with single-cell RNA-seq, bulk RNA-seq, multi-parameter flow cytometry, and also whole genome bisulfite sequencing, which is in here. So really took a systems biology approach to understand what was going on with these cells. Essentially, just for sake of time, not going to go through all of the data or what are the sort of differences between the cell types, but I hope it is pretty apparent from the slide that the expanded cells are quite different from the freshly isolated NK cells. And I'm not showing the sort of transcriptional pathway analysis, but they are very activated, very proliferative, very cytotoxic, and are expressing activating NK cell receptors. So great. We've had the good fortune of partnering with the clinical trial team for the Excel study. I would never have named this study because it has 800 words in it and no one is ever going to remember what's going on. But what it was and is basically the Hopkins myeloablative conditioning haploidentical stem cell transplant platform paired with 3 expanded NK cell infusions. And we were asked if we would be willing to do some of the correlative biology studies on the patient specimens as well as the product. And it made perfect sense that Megan was the right person to do that because she'd already been very deep into what was going on with the expanded NK cells themselves. So the diamonds are the points at which the NK cell infusions were given, and the K562 expressing the IL-21 were the feeder cells that were used to activate the cells. So essentially, we got our hands on the product, and we got our hands on biospecimens that were collected day 14, day 42, day 56, day 100, and NK cell infusions were given day -1, day +7, and then the number 3 column here is showing the 3rd infusion was meant to be given on day 42, but was given, kind of around that time, depending on the patient's clinical condition. Our goal was to be able to look at, the study ultimately enrolled 30 patients. We wanted to look at a matched number of patients that relapsed and patients that remained in remission. It's a pediatric trial, so it's taken a number of years for the trial to accrue. And so we knew that we were able to find patients who unfortunately had already relapsed. And so the pink and the blue are comparing those patients. Some of the upfront work that Megan did, which was really challenging, was to be able to isolate live NK cells from the biospecimens, because anyone who clinically cares for patients post-transplant, there aren't that many cells there at all, and so to be able to recover from a frozen sample that's been shipped all over the country, a viable NK cell was really tricky, but she figured out how to do it. And then here's some data from first blush. So we have a computational biologist who we've partnered with who's working closely with us in terms of how to integrate what we know clinically about the patients, biologically about NK cells in general, and then sort of the data that he's looking at. an A and B being patients that relapsed or patients that remained in remission, what we hoped to do was to combine the product with the biospecimens and then to sort of track the infused NK cells across the patient specimens as well, but weren't able to do that because the product was so different from the biospecimens. So essentially here on the left is the product. On the right, single-cell RNA-seq from the biospecimens from the 5 patients in remission and the 5 patients that relapsed. And then we're able to cluster and identify different NK cell subsets in each. And while I'll just trust me, I know standing up here, trust me, but on the left, the product is not very different. So regardless of whether the patients remain in remission or relapse, the product is, as Megan had found with the expansion, the NK cells are activated, they're highly proliferative, they're cytotoxic. We looked at their cytotoxicity against a K562 cell line in culture, and they all kill. So it's not like the patients who relapsed had bad products. But then on the right, looking across the biospecimens at the different time points, we are able to pull out some differences. And so it may be that what we're seeing is not related to the NK cells themselves. What we're seeing, meaning what happens clinically to the patients, is not because of the NK cells themselves, but probably is because of the host, because of the patient in whom the NK cell infusion is done. So to move on, I know I'm already talking way too long, but just to say, are expanded NK cells of a functional type? Yes, definitely. There is no question about that. And we are really excited about our emerging capability to look at clinical trial data, clinical trial biospecimens, and then really understand a bit more about what needs to be done to those NK cells for an effective therapy. What about CAR expression? This is very old data, so I'll just be very quick about it. But we started thinking about expressing chimeric receptors in NK cells because of the successes in T cells, but also with really deep thought about biologically what was happening in the NK cell and trying to be conscientious of the fact that a receptor that worked really well in a T cell was probably not going to be optimal for an NK cell. So this library of receptors is an initial library that we chose with intracellular signaling domains that we thought were really important for NK cell activation. And to cut to the chase, the 2b4 zeta CAR was the one that we found to be best in NK cells, both for expression, for stability of expression, for short-term cytotoxicity, and for persistence of the cells in vivo. The 4-1BB zeta CAR also works reasonably well, but at least in our head-to-head in vivo studies, the 2b4 zeta CAR seemed to work a bit better. So we, skipping, also years of preclinical study, I just wanted to get everyone excited about the potential for CAR NK cells as therapy, and to show that here on the top are 2 AML models, on the bottom are 2 ATRT as a pediatric brain tumor model, that the CAR NK cells infused targeting either CD123 or B7H3 have efficacy in these models, and efficacy that results in survival that is longer term of animals that are treated with NK cells expressing the CAR as compared to treatment with NK cells that are not expressing a CAR at all. So to answer the question, I think we feel like expression of the CAR is helpful, even though the NK cells obviously have their own activating receptors independent of the CAR. And the brain tumor work, I know I started out, I'm really excited about AML, and I've only been studying AML. I have a graduate student, Tony Cho, who also had a poster that I hope you all got to see, who came to the lab very jazzed about studying brain tumors. And I was really resistant to that because I didn't have any models, I don't really have the clinical experience, and yet he convinced me by finding a neuro-oncologist partner that we would be able to do that. And what it has done for me is also to be able to see on tissue specimens, which we don't have as readily from the AML models, that the CAR NK cells that he infused into the brain tumor penetrate into the tumor and also persist. So these tissue sections are from the animals at endpoint, and so both the NK cell treated and the CAR NK cell treated animals had disease and died of disease. But at the same time, it seems like if you have, again, some optimism and imagination, that the CAR NK cells penetrate a little bit better, and there are more of them than the regular NK cells. So Does expression of the CAR benefit? Yes, definitely. And I very strongly think that there is enhanced anti-tumor effect and localization, although I acknowledge I haven't shared all of that data with you. So just, again, trust me. So what's happening now? We are working very, very hard for our products to get to translation. That's a huge, heavy lift. In the meantime, this is as of yesterday or the day before, I can't remember when I made the slide, but on clinicaltrials.gov, for pediatric cancer, there are around 13 or 14 open clinical trials that are accruing across the country. So these are for both blood and solid tumors, exciting. They include a variety of different, again, activation, isolation, expansion methods for the NK cells. And at MD Anderson, they have their umbilical cord blood-derived NK cells expressing CARs and secreting IL-15, which are products that they have been using for some time. So really exciting. The future is bright. So we're hopeful for stability of funding. We're always hopeful for more funding, but we are sticking with it. And I think we're really excited to continue with the study of NK cell immunotherapy. Dahlia is ready to yank me off. Thank you all very much.
Thank you so much, Challice, for the amazing presentation. It's so exciting to hear about CAR NKs. I have to admit, I tried to secretly recruit one of your people, so next year you'll see posters on NK cells from my team. Okay, I was trying to give time for the microphone runners for questions.
So there were these recent reports of CAR cells transforming and forming tumors, rare, but do you have similar events happening in the NK field? And do you build in kill switches in your NK cells?
We have not put any kill switches in the NK cells. We certainly could. We haven't because we have not, and Natalie Hall, who presented last year and who had a poster this year and has been really focused on trying to get the NK cells to last longer. We haven't seen the NK cells persist long enough, honestly, to be worried about it. And that even in a profoundly immunosuppressed animal. And so you can imagine in a patient who has the capability to reject, they would last even less long. In terms of what about sort of insertional oncogenesis, I think the number of patients in whom that has been identified for CAR T cells is very small. Whether or not the sort of malignant disease that is identified after the CAR T cell therapy is because of the CAR T cells, then it becomes an even smaller number because there are some patients in whom they develop sort of a secondary malignancy regardless of the CAR T cell. So that is not something that I have personally been focused on because I think it is not as much a problem as the cancer itself. So I'd rather cure more of the cancer and then suffer maybe the 1% or less of the consequence.
I have a great talk. I have two questions. The first is, do you think there's a very big difference in the NK cells that were not part of the product in patients that were responder, non-responder? So they're host, well, they're all host, but yeah, the host NK cells, because you're using allogeneic in those, right?
Yes. So the study, the Excel study, again, I don't know all the words that are in the whole title, but the Excel study is donor-derived NK cells, same as the transplant donor.
Right.
So one of the things that we really, really hoped to do was to be able to track the infused NK cells as compared to those that were recovering post-transplant. And we sort of went back and forth. Can we use mitochondrial genes? Can we use some variants that are carried through in the clones that are not consequential to protein? But we cannot. The short answer is we cannot distinguish. So I don't know.
Yeah, is there a movement toward tracking them then, or like some way to know?
If
Like,
they were
can you
genetically
label them?
modified.
Yeah, yeah, okay.
Yeah.
So the CAR you'll be able to, CAR NK.
CAR you'll be able to,
Yeah.
or if it was a third-party
Yeah.
NK cell donor, you'd
Then
be
you
able
would
to,
be able
because
to.
presumably
Yeah.
the HLA would be different. But at least on this trial, they were the same as the transplant donor.
And what do you think is the biggest barrier to those patients having efficacy or not in terms of the fact that the product was so good?
I think the disease.
Yeah,
So
okay.
I really think it's probably something inherently resistant about the leukemia itself that either allowed it to survive or specifically was resistant to the NK cell cytotoxicity or killing.
Thanks so much.
Yeah, I'll ask you one. So one of our biggest enemies in the CAR T world is the microenvironment. I know you mentioned in your brain tumor models the mice did die from disease. So what are your thoughts about how resistant the CAR NKs are against the microenvironment, and what are your thoughts about combining CAR NKs with CAR Ts?
Yes. So Dahlia is an inspiration because of her work in really advancing how important it is to use immunocompetent models to be able to study some of these things. So Tony's brain tumor models are completely artificial because they are an NSG mouse, and then he puts in the tumor, and then he puts the NK cells either ICV or directly into the. So we don't have any of the microenvironment that we would otherwise have. That being said, we're trying really, really hard, Tony, thank you for all of your hard work, to engineer murine NK cells. So to isolate, and I can't even tell you the poor mice and the spleens and the number of NK cells, but to figure out how to do that so we can answer that question. I will say it was really nice to see in his tumors the infiltration of the NK cells, because I'm not sure that that was something that I expected. But I think if we knew what the problem was, we can engineer around that. We can solve that. But right now, we don't even know.
If there are no more questions, please join me in one more round of applause for Challice. Thank you so much.